So we've spent some time in this class discussing the importance of things like mitochondria and, well, the importance of things like, or the importance of mitochondria is that it allows us to fully break down sugars and convert the energy in those bonds into ATP. We also talked about the importance of chloroplasts, and the importance of chloroplasts are they allow plants to take light energy and create the sugars that other animals and plants are going to use to make a whole bunch of ATP. And so those structures, the mitochondria and chloroplasts, are pretty unique within the cells. And I'm going to talk about how they got there. So in the interest of not burying the lead, what endosymbiotic theory says is that proto-eukaryotic cells engulfed bacterial cells, and those bacterial cells allowed those eukaryotic cells to utilize oxygen so that they could have enough energy, and they allowed other eukaryotic cells to actually create sugars. So we're going to talk about the evidence for this and why it is why that particular evolution happened that way. So here's a cool little image of an electron microscope that just took a picture of some mitochondrial cells within a lung. Mitochondrial cells have two membranes, and we're going to talk about how that probably came to be as well. So here is a very basic image of, this is the book showing how endosymbiosis likely took place. So it says the first eukaryote may have originated from an ancestral prokaryote that had undergone membrane proliferation, compartmentalization of cellular function into a nucleus, lysosomes, and an endoplasmic reticulum. So that's this thing here. It's got membranes for some of its organelles already, so it is a eukaryote. Then it says in the establishment of the endosymbiotic relationships with an aerobic prokaryote. So again, that is a prokaryote that's able to use oxygen. So what these guys are teaming up so that this eukaryote can provide those bacteria with some protection, and those bacteria can provide that eukaryote with the ability to use oxygen. Continuing, and in some cases, a photosynthetic prokaryote to form a mitochondria and chloroplasts, respectively. So in the case of the chloroplasts, it's allowing the eukaryote to make some sugars, which is the origin of plants. In the case of the mitochondria, it's allowing an organism to be able to use oxygen to break down those sugars fully and make the most energy possible. And by the way, in my graduate work, I studied animals, I studied butterflies, and we studied in part their mitochondrial genome. So I'm going to be just discussing mitochondria because that's a little bit more about what I know about. But this idea also very much applies to chloroplasts as well. So this first picture here just kind of gives you a sense of, here's what the size of a bacteria is, here's what the size of a mitochondria is. They both have DNA. The DNA of a mitochondria is smaller than that of a bacteria. But the interesting part, I think, is that your mitochondria actually have separate genomes from the rest of your cell. It is, again, much smaller, but mitochondria have their own ribosomes. They're making their own proteins that are unique to the rest of your cell, which is kind of like the first piece of evidence that these things were separate bacteria at one point. As a bit of an aside, you get your mitochondrial DNA from your mother. So you and your mom have the same mitochondrial DNA. And she got her mitochondrial DNA from her grandmother. So it is passed on matrilineally. So be thankful to your mothers and grandmothers. That's where they gave you the powerhouse of your cell. So just thinking about a general size comparison, bacteria and mitochondria are very, very similar in size. This is a bit of a generalization. Bacteria actually can get quite large, but in general, this is the size range where you're going to find a vast majority of them, and you can see they're very similar size to your mitochondria. The next kind of piece of evidence that we have for mitochondria and chloroplasts originally being separate bacteria is that they replicate in a similar way. So most of the organelles in your cells are going to be sort of split up and divided equally between two sides. Your mitochondria are actually going to replicate their DNA and split in a fashion very similar to the binary fusion of bacteria. Same size, similar mode of replication. Their genomes look similar to one another. So in a mitochondrial genome, you're not going to have plasmids like you do in a bacterium, but you are going to have these small circular segments of DNA, which again look just like the ones that you have in bacteria. So the last sort of piece of evidence is that it's thought that these bacteria were absorbed into the eukaryotic cell using phagocytosis. And the phagocytosis, if you remember from before, what's going to happen is something is going to come up to the edge of the cell and then it's going to be engulfed. And once it's engulfed, it's going to be inside of a membrane inside of the cell, which is most likely where the double membrane of the mitochondria or the chloroplasts came from.